Okay, it is my pleasure to introduce Xinyou Yin. Well, actually, I got very inspired by your work during my PhD because, well, he was together with Martin Kropf, one of the first ones to propose this idea of linking QTLs and crop models to understand G by E. So it's really an honor to introduce you today. So the floor is yours.
Yes, thank you very much, Daniela, for introduction. And I would also like to thank Fred for kind invitation. Yeah, actually, it's my great honor to give this presentation in this symposium. Yeah, I would like to say, yeah, I saw Francisco is there. Yeah, Francisco is my co-author. Yeah, he's an expert in crop sensing. The other co-author is Dr. Wang, a former PhD student who graduated early this year. Yeah, this is how it works. Yeah, to talk about physiological crop models, I would start with the legacy left by Professor Keith DeWitt. Yeah, he already started to model the canopy photosynthesis around 60 years ago. Yeah, with his leadership, a number of crop models have been developed over the years in Wageningen. Yeah, this, by the way, diagram even did not include the models for greenhouse crops. Yeah, that everything will come tomorrow. Yeah, and you can see with his leadership, a number of crop models, especially those models ended with a cross has been developed. Yeah, and since somewhere early 90s, sucrose model has been used more or less as standard model for different applications of the modeling study. Yeah, one expectation is that the model could be used as could be a useful tool in analyzing genotype-to-phenotype relationships. And this is because the models using a number of input and translate this input to predict a number of output variables like yield, protein content, and resource use efficiency. Therefore, model was expected to analyzing so-called G by E by M interactions, and in which crop model input parameters are potentially be able to be subjected to the genetic analysis. One of the very early examples that we did is to map the sucrose model input parameters using the body decomponent input line populations. And you can see this table shows the Xinyou mapping result for the model input parameters. And the last column of this shows the percentage of phenotypic variation accounted for by the QDOs identified. And you can see for some input parameters, the percentage explained was higher than the percentage for other parameters, especially like this so-called partitioning coefficient. And so the next step is to test the model. And here it shows the model performance in predicting the difference of the yield among the individual lines of that population, either using the originally measured model input parameter values or the QDO-based model input parameters. And you can see model, performance was quite moderate, explaining only about 38 of phenotypic variation. What is the reason for that? Part of the reason, I think, is due to the error in measuring of the input parameters. And here it shows that the partitioning coefficient of the assimilate among different growing organs. To get this partitioning coefficient for different genotypes, we needed to do frequent periodical destructive sampling of plant materials to determine this coefficient. And you could imagine the data could become quite noisy, due to often using small samples. And this process is also time-consuming, and therefore it is not very amenable for large number of genotypes in a genetic population. Therefore, we defined so-called GWAS model, in which the model parameters are defined in such a way that they can be easy, that they are easy to measure. And also model structure was designed in a way that account for the interactions among interacting components, such as between carbon and nitrogen, between root and shoot, between source and sink component. So we're using the same data that we used earlier for testing sucrose to test GWAS. And you can see at least GWAS model can better explain the yield differences among two subgroups of the population. The difference in the two subgroups largely due to the segregation of a major gene in that population. But within a subgroup, the model performance is still a difficult one. So later on, a PhD student of ours, Nitim Kadam, used the model to analyze the yield in connection to the model input parameters. And you can see here, this slide shows the SNPs identified for individual parameters in comparison to the SNPs identified for crop yield itself. And you can see that a large number of SNPs has been identified. But in this context, we also test the GWAS performance in accounting for the yield differences among individual genotypes. And we use the data of 2013 for training, for determining parameters of the model. And you can see model explained about almost 60% of the phenotypic variation for control treatment and around 40% for the water deficit treatment. But when these parameters are used to predict the model in an independent year, in the following year, and you can see the R square become much lower. So we could argue that probably some of the model input parameters do not contribute to the model in explaining the yield differences. And I would like to show the example that from the work done by Junfei Gu, a former PhD student, in mapping the underlying biochemical parameters that determine the genetic variation of photosynthesis among so-called integration lines. And these parameters were determined using the gas exchange data for CO2 and light response curves. And then these parameters could be mapped in different genomic positions. And here we showed that the model performance in predicting the yield differences among the integration lines. And you can see that the model performance was again quite moderate for explaining yield differences either for well-watered or for drought spread treatment for yield or for biomass. But even this case, it is the simulation without using the genetic variation for photosynthesis. When genetic variation of photosynthetic parameters was included in the model, you can see the model performance even become worse, indicating that genetic variation in photosynthesis did not contribute at all to yield differences in the population. Okay. So then the next step is that modern crop sensing technology help to reduce the uncertainty of crop modeling in predicting G/E/M. And we know that crop growth involves a number of processes along both spatial and temporal scales, and also equivalently a number of sensing technologies available for different scales. and generating layers of information that could be used for modeling. And our expectation is that some data-based model information could be used as input to physiological clock models so that the model can be a better tool for predicting G by E by M. Yeah, this is illustrated by the work of Mr. Wang Dong, Dr. Wang Dong. He using this kind of technologies to predict both leaf and canopy nitrogen content using so-called machine learning techniques. Yeah, and using these techniques, he could predict the SLN and canopy nitrogen content based on different combinations of these crop sensing figures. And then the next step is to whether this kind of information could be incorporated to the clock modeling to reduce the model uncertainty. And in this case, so-called data assimilation technique could be introduced, in which, yeah, actually the simulation was updated at each step so that the model can better predict what is happening in the next step. And Mr. Wang considered two data simulation scenarios. One is using the observation from field measurement. The other is from the observations from remote sensing, yeah, predictions. And this was compared with the baseline simulation without data simulation technique. And you can see the prediction uncertainty was considerably reduced. So far, this has been done for M element. Yeah, we hope is that this approach could be also used to predict the G by E element, and hopefully it is successful. But a statement here is that even if it works, as a crop physiologist, we believe crop models remains an important tool because of its unique feature in heuristics. I just using one example for this, for QDL mapping of the for QDL mapping of SLA. SLA was measured 6 times during the growing season in this population. And we know that the flowering differs among individuals in this population as indicated by a magic QDL at this position on chromosome 3. Yeah? And when SLA measured at particular times was subjected to QDL analysis, the QDL profile peaks at different measurement times, except at flowering, yeah, indicating probably the existence of the QDL for SLA. But if SLA was collected by using ecophysiological models for some physiological age or developmental stage, and you can see that the peak of QDL profile identified in the first instance, they all disappeared, indicating that what we have seen in the first instance actually is the artifact of the flowering time QDL. Yeah. So with this, I have some summary point. I have showed that when crop models are confronted directly against G by E by M on yield observed in a genetic population, yeah, model's performance is modulated at best. Therefore, it is very useful to test an integrated approach in which some uncertain subjects, instead of being modeled, but are rather retrieved from in situ crop sensing in combination with the statistical techniques. Yeah? And the challenge is to see to what extent these modern sensing technologies in combination with the data simulation could detect subtle difference among genotypes in a genetic population. Yeah, we believe that data-based method will help this, but even overtake crop models in terms of prediction. But unique heuristic figure of the crop model remains crucial for unravel genotype-to-phenotype relationships. Yeah, with this, thank you for your attention.
Well, thanks, Xinyou, for bringing these very nice points about data assimilation and this main effect versus predicting within the environment aspects. Is there any quick question? Fred.
Yeah, I was wondering, if you look at the literature of the last 10 years, there are a number of groups that very strongly insist on genotype-specific parameters being some kind of physical constant that never change. Now, you show that, let's say, your model, well, not your models, but the classical crop growth models don't do very well when they are confronted with real-life data. I was wondering to which extent many of the parameters that are now inside the crop growth models actually have been tested to be genotype-specific, or let's say, or to be constant, because I think that if you look at what these parameters would do across different conditions, they are probably not so unshakably constant as is assumed.
Yeah, that's a very good point. And yeah, at the moment, we are sure that some of the parameters are quite stable across the environment, while others are less. Yeah. And yeah, I mean, when you get for those less stable parameters, multi sensing technologies in combination with a statistical method could be helpful. Yeah.
Thanks. Is there any burning question? Thanks again, Xinyou, for the nice talk.
Yes, very much.